All right, hello everyone. In this lecture, we're going to be talking about the epidemiology of vector-borne, or really more specifically arthropod-borne diseases. We'll start out by talking about vectors and then the transmission cycle and different modes of transmission, and then kind of wrap things up talking about surveillance and some emerging infectious diseases. First of all, a vector does not transmit disease. What it does is transmit a pathogen or parasite that can cause disease. So a pathogen is any disease-causing organism, and a parasite is an organism that lives on or in a host and obtains nutrients from said host. Now, ectoparasites live on the external surfaces of a host, whereas endoparasites live inside of their host. Obligate parasites cannot survive without their host, whereas facultative parasites tend to have both free-living and parasitic life stages, or some plasticity in terms of their lifestyle. And actually, that's it. Parasitism is a lifestyle. So it is not necessarily disease-causing. Many parasites will cause disease. Others may have minimal impacts on their host. So anyways, parasitism is this lifestyle, so viruses and bacteria are technically included in this definition, right? Living in or on their host, obtaining nutrients from their host. But in practice, viruses and bacteria are usually considered just pathogens, whereas protists, helminths, and other animal parasites, they're the parasites. So we do tend in practice to kind of separate out bacteria and viruses into the pathogen category, and then protists, helminths, and other parasites into the parasite category. Now, both can cause infection. Both parasites and pathogens can cause infection, and disease is defined as having clinical symptoms, some kind of noticeable negative effect on that host. Now, a host can be infected without actually being infective. Infective hosts are actively shedding pathogens or parasites back into their environment. They're capable of infecting others. So biological vectors, what they need is usually at least 2 blood meals. A first blood meal to acquire the infective agent, and a second blood meal to transmit it to some other host, right? They're a vector. Now, this lecture is predominantly going to be about biological vectors. In contrast, we do have mechanical vectors as well. Through mechanical transmission, typically through contaminated feet, mouth parts, anything like that, we will still have the spread of different types of diseases. But it is not going to be quite the same. There's no life cycle, there's no reproduction inside of the vector itself. Now, vectors aren't all bad. And this is the image that I have here, some of the other ecological roles of arthropod vectors in a variety of different ecosystems. They aren't all bad. There are many other important roles that they can play in an ecosystem. Being a disease vector is just one of them. So I want to broaden things out, not in terms of One Health in this case, but in terms of ecosystem function and this kind of broad diversity of their roles in different systems. So in terms of the transmission cycle, vector-borne agents are pathogens and parasites. They're highly adapted to living in invertebrate and vertebrate body systems. And these are very challenging environments, mind you. We need the parasite to, first of all, enter the host in some way. Locate, recognize, enter this host organism. Then find the right microhabitat within the host, whether that's in the blood, whether that's in the gut, whether that's in the lungs, wherever it may be. Then maintain its position, right? Battle off various host defenses, whether that's digestive enzymes or in the immune system itself, withstand environmental extremes. In vertebrates, this includes pretty high body temperatures. In any case, usually they're pretty oxygen-poor anaerobic environments too. Also, parasites and pathogens do need to try to avoid killing their host, at least until they've achieved their reproduction, they've actually followed through with their reproductive cycle. So then, that's not even the end of it. Then they need to actually pass on their genes. They need to get that next generation into some other host. So these are very, very difficult, challenging environments. And I think it's worth kind of recognizing not the skill, right? It's just evolutionary adaptations here that are involved. So beyond this, many parasites, protists and helminths specifically or especially, have very complex life cycles with separate life stages in different hosts, just adding more complexity to this process. Now, an intermediate host for these organisms is a host in which they are reproducing asexually. The definitive host is where the gametocytes come together and they're reproducing sexually. So, to persist in an environment, it totally depends on the accessibility, the susceptibility, and the transmissibility regarding their vertebrate host. In terms of accessibility, we're just saying, hey, hosts are abundant and they're available to vectors in the environment. And this availability, it might be seasonal, so in terms of just synchronized phenology, or it could have to do with daily cycles. As far as susceptibility goes, the hosts do kind of have to permit infection in a way, right? The parasites or the pathogens need to typically be maintained in the peripheral circulatory system for long enough for them to complete their development and without, the host succumbing to illness. And then transmissibility. This is where suitable hosts need to stay in the population. The parasites or pathogens need something to reinfect at some point. There needs to be somewhere else to go. So for suitable hosts to remain abundant, this might mean that just immunity is rather short-lived, or there's very short generation times for those hosts. Let's look at a generalized transmission cycle for an arbovirus, in this case Japanese encephalitis virus, as a just simplified example of the kinds of cycling we can see with vector-borne disease. So in this case, we've got the vector right here in the center, Culex mosquito, and we normally have cycling within this enzootic or sylvatic cycle, which is just among wild animals, in this case, wild water birds, where they're acting as a reservoir. And with these reservoir hosts, they're very important for maintaining the pathogen in the environment. This is called the enzootic cycle within animals. That's literally what that word means. So these animals, these birds, they can be infected for very long periods of time, but not develop a lot of symptoms. So outside of this, sometimes we do have spillover into a secondary cycle called epizootic cycle. And in this kind of cycle, we see a different type of host, an amplifying host. Hosts that aren't normally exposed to this kind of virus, they're going to be a little bit more susceptible. This is an acute infection. The infection period is much shorter, and they're going to exhibit symptoms of this disease. So in these amplifying hosts, suddenly we've got a lot more input of viral particles of the pathogen in this system here. And because of their proximity often, because this is a pig, this is livestock to humans, this proximity to humans, we will often see a little bit of crossover. So in this system, when humans are infected via a bridge vector, because they're a dead-end host, humans, other organisms that aren't good hosts for this virus, the virus either it starts to kind of infect the organism, it can't be fully replicated, there's some issues, it's never transmitted. These are dead-end hosts. There's no cycling out of these hosts. If you were to get bit by a Culex mosquito again, they're not going to pick up that virus and move it back into one of these cycles. These are incidental or dead-end hosts. So, like I said, this is just a simplified example showing us this enzootic or sylvatic cycle, the epizootic, this kind of spillover amplifying cycle, and then how we can have this sort of tangent out via a bridge vector into incidental or dead-end hosts. Now, as you can imagine, there are a variety of ways that a vector-borne disease can actually be transmitted. And it's not just through the bite of the vector itself. First, let's talk about horizontal transmission, which is from vector to host, or I guess maybe I should say from host to host via a vector. So most common is anterior station transmission, which is where the vector transmits the pathogen or parasite while feeding from the mouth parts or the salivary glands. For example, mosquitoes transmitting malaria or filarial worms. With posterior station transmission, this is transmission via feces or frass. So the vector eliminates on the host, the bite is itchy, and the host ends up scratching this infectious agent from the feces into the skin. Examples of this would be Chagas disease or louse-borne epidemic typhus. Next is accidental transmission, and this isn't about the route through the vector, it's more about the choice of host, and it's an unintended host. Well, I mean, it's unintended at least from the perspective of the pathogen or parasite. These are hosts that can't sustain the agent, or they just can't become infective, which leads to a dead end for the disease. So in this case, we call the vector a bridge vector, since the pathogen or parasite was transmitted tangentially via a bridge to an incidental or dead-end host. Now, okay, this next one isn't on the slide, but I did want to mention multiplicative or propagative transmission, which is where the pathogen actually, well, replicates / multiplies within the vector itself. This is very common for many arboviruses, and remember, in this case, with multiplicative or propagative transmission, it's the number of pathogens that changes, not the form. So then we move into vertical transmission. And with vertical transmission, did I not mention mechanical? Oh, I'm sorry. So with mechanical transmission, this is via contact. There's no sustaining of the pathogen within the anatomy of the vector. It is strictly, I don't know, inanimate, right? But it's still an organism. So, with vertical transmission, now the definition does depend on the focal organism, whether it's the host or the vector. If we're looking at the host, which is what I'm going to give you first, well, it's going to be simple transmission from parent to offspring. So that's it. Now, if we're looking at vertical transmission from the vector's perspective, the definition is, well, vector to vector transmission. First is transstadial transmission, which is just where an agent is passed from one life stage to the next through successive molts, or maybe transgenerational, so from parent to offspring, but this is usually transovarial, so from the mother. And then finally, rarely, and I do need to emphasize rarely here, there may be transmission through direct contact venereally, so from male to female or vice versa during mating. Vectorial capacity refers to or quantifies the transmission potential of a vector-borne pathogen or parasite in a susceptible population. And this concept of vectorial capacity considers ecological factors as well as the behavior and life history traits of the vector itself. Mathematically, this is the equation or kind of standard generalized equation that conceptualizes or quantifies vectorial capacity. So first, let's look at C. C refers to that vectorial capacity, and it's equal to, first, the density of the vector itself, represented as m, then the biting rate of the vector, A, the daily survival rate, P, the incubation period of the agent, N, vector competence, B, and with vector competence, we're talking about the ability of the vector to first acquire and then transmit the agent. It's easy for a vector to become infected with some kind of agent, but to be infective, to be able to transmit that agent, does take, well, we've already talked about how complicated it can be to be a pathogen or parasite in the world today. Okay, so you understand that. And then finally, we have 1 over the negative natural log of P, and P was daily survival rate. This is just looking at lifetime survival of the vector. So when we look at this equation, there's a lot of stuff we can pull out of it. First of all, vectorial capacity is absolutely density dependent, right? Vector density M was a term in this equation. So the more vectors, just the more mosquitoes in this case, right? You have the greater vectorial capacity there is for this vector-borne disease-causing agent. The other thing I really want to note is that 2 bites are required. We've already talked about that, right? We need to take not 1 but 2 or more blood meals to first acquire and then transmit this disease-causing agent. This is why we see A squared in the equation. So the vector needs to first acquire the agent and then, by the way, survive. It needs to survive the extrinsic incubation period, EIP, which is quantified in days, typically, to then transmit upon a second bite this disease-causing agent. And there's a lot of stuff that we can pull out from this equation. For example, looking at the term P to the N, right, taking that pathogen, taking that daily survival rate and raising it to the power of the pathogen incubation period, this tells us, well, the probability that the vector, in this case a mosquito, it survives through this extrinsic incubation period to actually be able to transmit an agent with that second bite, right? A squared. The other thing that we can pull out from this, I suppose, is maybe MA, very conceptually, which is just going to be the total bites to a host or potential host, likely a human, per day. Now, this equation, I think, is very useful for quantifying the potential transmission of various diseases. Finally, surveillance. Now, in the United States, led by the CDC, the Centers for Disease Controls and Prevention, is going to be the National Notifiable Diseases Surveillance System, NNDSS. I'm not sure if people shorthand to this to like N2DS2 or like NNDS2. People shorthand things in funny ways. Anyways, with this system, it includes many vector-borne illnesses, and of these, mostly mosquito or tick-borne illnesses, including anaplasmosis, a lot of arboviruses like Eastern equine encephalitis, or maybe the Powassan, St. Louis encephalitis, or even things like lacrosse encephalitis, plus dengue, Lyme disease, plague, right, and Zika virus. Now, reporting is mostly based on what's called passive case detection, and this is true for both human and veterinary cases. So only individuals that are looking for medical attention and then properly diagnosed and with disease confirmed with laboratory testing, like a seropositive blood test, are going to be recognized within this system. And with serological testing, we're looking at antibodies in response to the infectious agent. However, with many vector-borne illnesses that, we're dealing with, a lot of them have non-specific systems, elevated temperature, headache, fever, what have you. So a lot of them will be missed, and ultimately disease prevalence is going to be underreported. Now, the World Health Organization, right, the WHO, they today only require countries to report events of potentially international public concern. In the past, if we're considering arthropod-borne diseases, they only cared about yellow fever. There's other things that they care about, of course, but in terms of arthropod-borne diseases, in the past, the WHO has only cared about yellow fever. Okay, actually, the last thing I wanted to talk about here were emerging infectious diseases. And by this, I mean any kind of new or reemerging known disease with increased incidence or broadened geographic range. And the reasons we might see these diseases increasing in their prevalence is through a variety of factors, like population growth and density. With increasing population growth, increasing density, we of course see increased contact between potentially infected individuals, human-animal interactions. These zoonoses, right, jumps from the sylvatic cycle to potentially human hosts, urbanization and globalization spread throughout the globe, interconnectivity, as well as concentration of human bodies. Other factors like antimicrobial resistance, climatic change, very important, not even on this list, poverty and war, bioterrorism, all of this, very important. Emerging infectious disease, anything increasing in prevalence since the 1940s, every decade, increasing since then. Now, arthropod-borne emerging infectious diseases include things like Lyme disease, tularemia, which is rabbit fever, West Nile virus, yellow fever, and Zika virus. Related are something called neglected tropical diseases. So with neglected tropical diseases, this is essentially anything except for HIV/AIDS, tuberculosis, and malaria. Those are the big three, you might call them. So many resources go towards those three diseases: HIV, tuberculosis, and malaria. With neglected tropical diseases, which altogether have, if anything, more of an impact than any one of those, they're very common, especially in low-income populations in developing regions of Africa, Asia, and even the Americas. So many of those neglected tropical diseases can often be, I guess, plagued by not a lot of symptoms, right? Asymptomatic, not a lot of symptoms, longer incubation periods, a lot more time between exposure and whatever the end result is. There's also a lot of geographic isolation, there's a lot of clustering, endemicism, which just honestly makes treatment and prevention so much more difficult. So examples of arthropod-borne neglected tropical diseases include dengue, African trypanosomiasis, which is sleeping sickness, lymphatic filariasis caused by helminths, nematodes, and then onchocerciasis, or river blindness. So that's everything for this lecture. If you have any questions, as always, please let me know.